All right, so now let's start talking about growing microbes in the lab. So, a culture medium is a nutrient material prepared to grow microbes in the lab. A couple of things that we need to be absolutely certain about if we're doing that. First is that it's sterile. There can't be any microbes in it, right? Because if we are growing, let's say E. coli in the lab, and our culture has Klebsiella pneumoniae in it, then anything that I'm seeing as a result of the experiment that I'm doing, is it because of the Klebsiella or is it because of the E. coli, right? So I need to make sure that my culture medium is sterile. Then we have the idea of inoculum, right? So if I have a sterile medium and I am now adding that microbe into the medium sterilely via inoculation, and then culture is when the microbe has grown in or on the culture medium. So agar is a complex polysaccharide used as a solidifying agent for culture media. So these would have been the things that you used in lab. So these are Petri dishes, and also known as Petri plates, in which media is put in. And here you can see a slant, and you can see why it gets that name. And here you can see a deep. And each of these is useful for different reasons for growing bacteria. But generally, the reason we use agar is because it solidifies the media, and bacteria don't typically eat it. So it liquefies at 100 degrees Celsius and it solidifies at about 40 degrees Celsius. So it's a perfect material for culturing bacteria. And we have two main types of media. So we have a chemically defined media. It's a media where the exact chemical composition is known, right? Now, I don't need you to know moles and how to calculate moles, so don't worry. But I need you to think back to when you learned about moles, right? If I told you I have 5 grams of glucose, right? And I told you this is the formula for glucose, right? The chemical formula. Therefore, here's the formula weight of glucose. You could figure out how many moles of glucose you have, right? That's all the moles you need to know for this, all right? So chemically defined media, I know molecule for molecule how many molecules of glucose there are, how many molecules of this there are, etc., right? So that is a chemically defined media. I have a really good idea of what's in the media. Okay, so this is a chemically defined medium to grow Escherichia coli. Now, we also have what's known as complex media. So complex media is made from extracts and digests of yeast, meat, or plants. So here you can see we have peptone, which is a partially digested protein, 5 grams, beef extract, 3 grams, sodium chloride, 8 grams, agar, 15 grams, and water, 1 liter. So the sodium chloride, the agar, and the water, you could probably figure out how many moles you have. But what is beef extract? How many grams of glucose is in there? How many grams of, methionine is in there? We don't know. And so a complex media is not that well. You don't know it on a molecule by molecule level. Complex media in a liquid form is called a nutrient broth, while a nutrient agar is the solid form. So you would have here Luria broth, LB, and Luria agar, LA, or typically in the microbiome lab at LaGuardia we use TSA or a tryptic soy agar, right, which would be in the plate. And we have TSB, or tryptic soy broth, which is the broth form of the tryptic soy media. Since anaerobic bacteria can be killed by oxygen, they have to be grown in a reducing media. And so we typically are going to put it in a place where it contains chemicals that combine with oxygen and or heat it up to drive off oxygen. So you can see here, a anaerobic jar. In this anaerobic jar, you have a catalyst of palladium, and you have an O-ring gasket to make it completely airtight. And then you have an envelope that contains sodium bicarbonate and sodium borohydride, and you have an anaerobic indicator. And so this creates a completely oxygen-free environment for your bacteria to grow in, or you can grow it in an anaerobic chamber. Now, typically, we wouldn't do this in a undergraduate microbiome course, but it is something that you might see out in the real world. Some bacteria we can't actually grow in culture. So an example of this is Mycobacterium leprae, which is the cause of leprosy. It does not grow on the cultures so far. So the only thing that it will grow on in a laboratory setting is on an armadillo. Okay, so just a really interesting example. So special methods have to be used to culture capnophiles. These are microbes which require high CO2 concentrations. Either we can put them in a jar with a CO2 packet, or we can put them in what's known as a candle jar. And a candle jar is exactly what it sounds like. It's a jar with a candle on it. And as you know, as you can imagine, a candle will burn so long as there's oxygen. The candle will go out once the oxygen level is depleted, and once the oxygen is depleted. We also have selective media. So, when you hear this term selective media, think of natural selection, right? Natural selection is, of course, has to do with evolution, but in this case, what we're talking about is selection in terms of the environment, right? So natural selection is certain traits are favored or more likely to survive in a given environment, right? So, we have the ground in a particular area is very dark, right? And a mouse that is light in its fur color is more likely to stand out against that dark background than a mouse that has a dark color, right? So if we have so that is a natural selection process, right? So selective media involves creating an environment that favors the growth of certain organisms over others. So an example of this is MSA, or mannitol salt agar. So here we have a very high concentration of sodium sodium chloride in the media, right? So the media therefore favors the growth of gram-positive organisms over, let's say, gram-negative organisms. So that will favor, Staphylococcus epidermidis and Staphylococcus aureus are both able to grow in that environment. Now, on top of that, we're going to add this medium contains mannitol, which is a sugar alcohol. It's actually used a lot of times to sweeten sugar-free candies, and phenol red, right, which is a pH indicator. So what happens is, you can see in the illustration, Staphylococcus epidermidis and Staphylococcus aureus both grow. However, Staphylococcus aureus is able to additionally break down that mannitol and ferment it. And when it ferments it, it creates an acidic environment. And that acidic environment interacts with the phenol red, turning it yellow. Then we have differential media, which makes it easy to distinguish colonies of different microbes. So notice the selective media actually prevented certain organisms from growing, right? MSA plate, because of the high salt content, favors the growth of gram-positive organisms and disfavors the growth of gram-negative. Now, differential media, on the other hand, isn't going to prevent anybody from growing, right? Instead, what it's going to do is it's going to create an environment that helps to tell these different ones apart. So an example of this is EMB. EMB contains peptone, lactose, dipotassium phosphate, eosin dye, methylene blue dye, and agar. And the lactose fermenters cause the dye to change blue, whereas an organism that does not ferment lactose remains colorless. And E. coli and Citrobacter and Enterobacter species, they do a pretty amazing thing where they turn the dye green. Now, in the illustration I provided, you don't really see that effect. Alas, I was unable to find a picture of this, but were you to work in the lab, you would actually see this metallic green look to this, to when these guys grow on EMB media. Then we have enrichment culture. It's a type of culture which encourages the growth of a desired microbe, right? Again, we're not keeping anybody from growing, but we are creating an environment that enriches the growth of one versus the other. So let's say I wanted to find, let's say I was looking for a bacteria that breaks down the chemical phenol, right? So I find a site where there's tons of phenol pollution. The phenol has been in the ground for a long time. Somebody was not a good environmental steward. So the phenol is now in that soil, polluted the soil. The soil is deadly, really, for most organisms. But I can also assume that in that soil there are some bacteria that break down phenol. So what I can do is I can take some soil sample and create a phenol-containing culture, right? So I have a culture tube, and in that culture tube, I have a phenol. That's the only thing that a bacteria in this environment can eat, right? I add my soil sample, and I incubate it. Theoretically, what's happening is there may be a few bacteria in that soil that don't eat phenol, but I'm not giving them anything to eat, but I'm also not killing them, right? And then what I'm going to do is I'm going to transfer. So I wait a while, the bacteria grow, then I transfer 1 ml from that original culture to a new culture, right? And that 1 ml will probably contain some phenol-eating bacteria. And in this new culture, I feed it more phenol. Again, I'm not stopping anybody from living, but I'm also not feeding them, right? And so then I do this process a few times, and after a few times, the only bacteria that will remain when I take something out will be bacteria that eat phenol. So, key difference between enriched media and enrichment media. Enriched media is like we're adding stuff like blood and other nutrients added to a general purpose media to encourage the growth of many types of organisms. An enrichment media promotes the growth of a particular organism. It provides essential nutrients for that organism, but because it's enrichment, not selective, it does not contain inhibitors of the growth of those competitors. And generally, we scientists, we like to work with something known as a pure culture. So pure culture contains only one species or strain. And a colony is a population of cells arising from a single cell or spore, or from a group of attached cells. And a colony is often called a colony-forming unit. Now, the thing is, why do we like pure cultures? because we want to avoid confounding factors. So you can imagine, right? I have a tube, and in that tube, right, I have two types of bacteria. And I'm going to make in this tube, one bacteria is a coccus, one bacteria is a rod, just to make it simple, right? And so now these two bacteria have two different metabolisms. They eat two different sets of things. So now I put these two bacteria together and they do something. Well, who did it? Was it the coccus bacteria or was it the rod bacteria? Which bacteria is it that has the effect? I would never know unless I had a pure culture. And so the streak plate method is used to isolate pure cultures. So you can see here, stroke one, so what you do is you would take your loop, you would dip it into a culture, and then you would spread it on that plate like seen in one, right? And then you would make stroke number two, and stroke number two would intersect with one at several points. So it would spread out the bacteria that's in stroke one. And then you would do stroke three, which would spread them out even further. By the time you're getting to stroke three, you should see individual bacteria, which we can then pick and say this represents a single bacteria with a single set of characteristics.